Hello everyone, my name is Andrea. I am the Regional Marketing Specialist from GenScript. Today's topic for the webinar session is Antibody Protein Engineering Solutions to Achieve Your Perfect Molecule. And I'm very excited to introduce you the presenter today, Dr. Grace Tan. Dr. Tan is the Product Manager of Reagent Antibody Services at GenScript. She graduated with her PhD in pharmacology from Stony Brook University, where she structurally characterized novel inhibitors for insulin-degrading enzyme, and also worked on developing a new approach to determine the substrate spectrum of an ubiquitin conjugating enzyme. Today's webinar will take around 45 minutes. If you have any question, feel free to type in the question field that you see on your screen, and we'll answering them after the presentation. If we didn't get to answer your question during the Q&A session, we'll be sure to take down and email back to you afterward. We'll record down this webinar, which will also be available on our website after today. Lastly, here is the list of upcoming webinars that we are rolling out on the following weeks. Without further ado, let's welcome Dr. Grace Tan to start her presentation. Dr. Tan, over to you.
Hi, Andrea. Thank you everyone so much for taking the time to join me today for this webinar. My name is Grace Tan, and just before I start, I just wanted to quickly introduce GenScript. For those of you that may not know us, GenScript is a contract research organization, and we specialize in providing services and products to help researchers like yourself get answers to your research questions quicker. We take pride in being the #1 gene synthesis provider in the world, with over 110,000 customers in over 160 countries. To help us achieve this, we have over 2,900 dedicated employees globally, with about 30% holding a graduate level degree. We not only make DNA, but we also do a lot more. We have comprehensive portfolios focused on customized protein services, antibody production, NGS panels, and peptide services. And we also offer a number of catalog products and services focused on the development of biologics. For much of the 20th century, pharmaceutical research has focused on the development of small molecules as therapeutic agents. However, in the past decade, with major technological advancements in research and development and increased knowledge of the intricacies of how biological systems work, the biopharmaceutical industry has increasingly focused on the development of large biomolecules as therapies for diseases. We refer to these popularly as biologics, and biologics are typically proteins, peptides, nucleic acids, or even cells. Biologics development is favored because these molecules are able to reach targets previously considered to be undruggable by small molecules, and they are also typically more specific to the pharmacological target compared to small molecules, reducing undesired medical side effects. However, seeing as biologics are considerably larger and more complex, developing them into therapies suitable for the clinic can be tricky. Take this molecule of aspirin, which comprises just 21 atoms, and compare that to a typical therapeutic antibody, which can consist of over 20,000 atoms. To identify, optimize, and produce the best antibody, protein, or organism for that matter is therefore no easy task. Given the complexity of developing the perfect molecule, a lot of scientists now recognize that perhaps it's time we need to approach biology differently. As a result, increasingly scientists have started to think like engineers and have adopted a radical mindset that began and actually now forms the basis of the field of synthetic biology. Synthetic biology adheres to 4 key engineering principles that you see on the right. Namely, these principles are design, build, test, and learn. And can be cycled through again and again until your optimal molecule is developed. This engineering mindset enables scientists to adopt a bottoms-up approach where the design, construction, and optimization of molecules and biological systems can be approached in a very rational and very controlled manner. An example of where the engineering mindset has really taken root and grown is in the area of antibody drug discovery and development that I will be covering today. In antibody drug discovery and development, there are many opportunities for using synthetic biology principles to engineer and identify the optimal antibody molecule. Let me take you through it. Once a target for which a therapeutic antibody is to be developed against is identified, the first step of the drug development process is antibody lead generation, which is shown right here in this box. At this stage, you want to be able to identify the best and most promising antibody candidate. There are several popular antibody generation technologies that currently exist to choose from, such as hybridoma generation, cell sorting, as well as display libraries. Next, once an antibody lead has been identified, you will need to optimize it. During the optimization stage, the antibody is mutated to generate a variant which is then expressed and screened. In order to introduce mutations at specific sites or regions within an antibody, researchers use a synthetic biology approach, primarily using bioinformatics to design a DNA mutant library. They will then use this library to generate expression constructs of antibody mutants and express these mutants at a small scale and screen them in a high throughput fashion. The purpose of these optimization steps is to introduce or enhance specific characteristics of the antibody candidate, such as improved binding, improved affinity, stability, improved expression, etc. And this lead optimization process can be cycled through multiple times to further optimize the antibody until it's what you desire. Finally, when you've identified the ideal candidate, you will move on to the larger scale antibody production to ensure that your target antibody can be expressed and purified at high yields, right here. Seeing this drug development schematic, you can imagine, therefore, that generating the perfect antibody and protein candidate is really not a trivial process, and there are a lot of risks involved. Some of the major fears that arise during the drug discovery and development process for scientists could look like missing opportunities, such as lacking the technologies to be able to generate quality lead candidates. This could result in not fully maximizing the number of leads you could have generated or the number of mutants you could have screened. And as a result, you could be missing out on a potentially groundbreaking protein or antibody lead and waste precious time and financial investment in the process. A second key setback would be for mistakes to occur during research. This could arise particularly for scientists tackling especially challenging projects and where limitations of in-house technologies prevent reliable generation, optimization, and production of quality lead candidates. Fortunately, GenScript has expertise to help scientists along this process, as shown here. Our services are incredibly modular, so researchers can really have full control over the drug discovery process and choose where within this process they would like to harness GenScript's expertise. So we support protein and antibody engineering research through our core platforms. On the far left, you see here, for lead generation, we have our award-winning custom antibody generation service, where scientists can work with us to develop a highly tailored antibody generation plan. We've generated almost 50,000 antibodies to date, and also recently acquired Berkeley Light's Beacon platform for single B-cell sorting. Next, for lead optimization, as I mentioned at the very beginning, we are the #1 supplier of gene synthesis in the world. We utilize next generation DNA synthesis and can make any gene in any vector. We offer guaranteed turnaround time and have over 10 years experience in library construction. Finally, for lead production, our unique high density protein expression system allows us to increase antibody yield 4 to 8 times with easy scale up options. And I just want to mention that while I'm focusing on antibody engineering for drug discovery and development in this webinar, I did want to say that our DNA and recombinant protein expertise and services do extend to protein engineering as well. So let us take a look at the drug discovery and development workflow. Here are some examples of typical challenges one can make during antibody lead generation. You might be missing good binders, you might not generate enough binders, or you might not you might have to select the best antibody generation method among the many that already exist. For antibody lead optimization, you might have poor coverage, your DNA library might be biased, or you could have poor library expression. For antibody production, you might be struggling with low yield, scalability challenges, and poor reproducibility. I will review the solutions that GenScript can offer at each of these 3 stages that I have highlighted right here. For antibody lead generation against a specific biological target, GenScript harnesses Optimum Antigen, a proprietary bioinformatics platform, to assist researchers in optimizing the design of antigens and ensuring that they have maximum immunogenicity. Additionally, we do offer animal immunization services to harness the animal's natural ability to generate good lead antibodies. In order to progress to the antibody lead optimization stage, monoclonal antibodies first need to be isolated and identified. Antibodies from animals can be produced from hybridomas, or alternatively, can be fast-tracked and identified more rapidly using single B-cell sorting. As mentioned in the previous slide, hybridoma technology has been the time-tested method for antibody discovery for the last 50 years. GenScript is really familiar with this technology, and we have generated over 8,000 custom MABs to date using this method. Excitingly, we do also now have the Beacon system from Berkeley Lights, which can greatly speed up the antibody discovery process by reducing the screening time from several weeks to just 1 day. By using the Beacon platform, plasma B cells from immunized animals are screened using multiple binding and functional assays to select only the most qualified antibody leads in 24 hours. We are currently recruiting beta testers to use the service for both rodent and rabbit projects, so please do get in touch if you are interested in trying this service out. You know, while GenScript can also generate antibodies from rodents, one key antibody generation technology I would like to highlight is MonoRab, GenScript's premier rabbit antibody discovery platform. Due to the rabbit's robust and unique immune system, rabbit antibodies often can be generated against challenging targets, even targets that are typically non-immunogenic in mice. Additionally, rabbit antibodies are typically superior in specificity and affinity to mouse antibodies. In the example on the left, GenScript produced a high-affinity anti-idiotype rabbit MAB against pembrolizumab, and a monoclonal antibody drug. From the ELISA experiment, we found that this anti-idiotype rabbit MAB had an EC50 of just 5 nanograms per mil. Rabbit antibodies are also great for immunohistochemistry applications. Here, we compare a mouse anti-beta-actin MAB here, highlighted in panel A in red, and compared it with rabbit anti-beta-actin, which is in green, shown in panel B. When co-localized, the resulting image revealed significantly more binding of rabbit anti-beta-actin than mouse at a greater intensity, as highlighted here by the white arrows. All this taken to indicate that the rabbit anti-beta-actin MAB had a higher affinity than its mouse-generated counterpart. After you identify a good quality lead, researchers can further work to improve the lead's physical or functional characteristics and properties. Thanks to synthetic biology, scientists can engineer a library of different mutant proteins and antibodies and screen them to isolate the best candidate. In the case of antibody engineering, you can screen different mutants to improve specificity and stability. You can mutate the complementary determining regions to improve antibody affinity. You can remove degradation hotspots and control thermal stability. For protein engineers, you can optimize enzyme function, protein thermostability, and also engineer new fusion protein formats. To accomplish this screening effectively, the rational design and synthesis of large DNA variant libraries is necessary. Fortunately, at GenScript, we are able to offer high-quality, high-throughput DNA synthesis services thanks to our third-generation DNA synthesis technology. Unlike first-gen column-based DNA synthesis and second-gen DNA printing on a silicon chip, We use a third-generation semiconductor chip technology that enables us to synthesize thousands of oligos on a single chip. This semiconductor technology, which is similar to the technology used to make computer chips, cell phones, and other electronic devices, enables us to reduce the bias in codon usage and poor variant representation that is commonly seen in current mutant library construction methods. The semiconductor chip we use is an array of hundreds of thousands of platinum electrodes on which we synthesize oligos used for mutant library construction. Each one of these electrodes can be individually and precisely controlled by a computer software during the synthesis process. An individual electrode can be activated and allows the addition of one nucleotide at a time. As such, we can synthesize unique oligos on each of these electrodes that we can subsequently use in the assembly of variant libraries. An example of how this precise control of DNA synthesis can be applied is through the generation of precision mutant libraries. To build a precision mutant library, we use chip-synthesized oligos that encode all amino acids of your choice at specific sites and stitch them to the constant region of the template. This allows us to create either a site saturation library that is delivered as a separate pool for each individual site, or a saturation scanning library with multiple sites mixed together. We can also assemble different mutation regions and create a combinatorial mutant library, which has multiple sites mutated simultaneously. We can check the quality of the library by using NGS to sequence the DNA. An example of an application of a precision mutant library is in the situation where you are looking to conduct affinity maturation of an antibody lead candidate. Often, researchers need to mutate multiple antibody sites with particular amino acid sequences based on their preliminary data and prediction modeling. With antibody affinity maturation, this would normally occur in the CDR region of the antibody because this is where binding to the target antigen occurs. In this case, we created a combinatorial mutagenesis library to improve antibody affinity. We aimed at mutating 4 sequential sites in the CDR regions of the antibody, and particular amino acids were desired at each position. For the first site, AA1, we wanted to mutate them to 3 different amino acids. The second site, AA2, to 10 amino acids, and 20 amino acids for sites AA3 and AA4. We utilize our semiconductor technology to synthesize the precise oligos and constructed the library. The distribution of the library as designed is shown on the left, and the NGS result is shown on the right. We can see that both the design distribution and NGS validation results are almost identical. If we compare the deviation of the amino acid ratio achieved from the designed ratio, it is less than 25% in most sites. We overall got 92.2% coverage for this library of 12,000 mutants. Additionally, to compare our precision mutant library with a traditional NNK library generation methodology, we made the same library with NNK oligos. On the left, you can see that for the first and second sites, even though we only needed 3 and 10 amino acids respectively, the NNK library did not have the ability to exclude unwanted amino acids. Furthermore, for all 4 sites, the distribution of each amino acid was a lot more uneven in the NNK library compared to the equal representation shown with our semiconductor-based mutant library. If we take a closer look comparing sites 3 and 4, the deviation of amino acid ratio from the design ratio is huge in the NNK library, with 77.5% larger than the 25% deviation, while the same statistics from our semiconductor-based library was much smaller. This indicates to us that using semiconductor-based oligos eliminates bias of amino acid distribution and increases screening efficiency. I also just wanted to say that our precision mutant libraries aren't just limited to antibody optimization. Here, I wanted to quickly show an example of how our clients use our precision mutant library in an enzyme engineering study to improve enzyme activity. The enzyme our client was trying to optimize had 484 amino acids, and they were interested in 3 regions around the enzyme active site for a total of 38 positions. Our client wished to create a saturation scanning mutagenesis library and scan all possible single mutations using 19 amino acids other than the wild type at these sites. This totaled 722 mutants, and from these 722 mutants, they wanted to find variants with improved activity. The NGS results showed an equal distribution of amino acids across all the sites with variants representing all non-wild type amino acids present at each site. Precision control of content has a lot of advantages. It allows you to take full control of what codon you need at each location without bias, with superior diversity and coverage, resulting in a much smaller library size with only the variants you want to test, minimizing screening burden and downstream costs. Just as a quick summary of the key benefits of PML is that it is able to eliminate sequence bias. All 64 codons can be used. It is able to prevent the use of undesirable motifs, allows codon optimization, and helps you avoid stop codons. All of these key benefits help you expedite downstream screening for your antibody and protein engineering projects. When you finally decide on an antibody or protein you want to work with, you have to produce them at sufficient quantities to use for various research assays. And when it comes to your desired proteins and antibodies, you also want to ensure that you obtain the amount you need in an efficient and cost-effective manner without compromising on the quality of the product. GenScript's protein services is run by a team of expert scientists who have over 95% expression success rate with more than 10,000 expression projects delivered to date. Part of how we've been able to be so successful in protein and antibody production is tied to our high-density expression system, or HD system. Our HD mammalian expression system uses a proprietary cocktail reagent that is introduced post-transfection in cell culture media for transient expression. The cocktails we use are tailored to specific antibody formats and are able to boost cell growth, regulate cell cycle, and have been shown to achieve 20% higher cell viability. For example, cocktail 1 in yellow is best suited for expression of human IgG1, mouse IgG2A, and rabbit IgG, and has been shown to increase antibody titer 2 to 6-fold. Cocktail 2 in blue is best suited for mouse IgG1 and has been found to increase antibody titer 2 to 4-fold and facilitates the folding and secretion of this antibody format. Here, we show that our HD system outperforms other standard expression systems by our competitor. When we expressed 10 different recombinant monoclonal antibodies, we found that compared to our competitor, GenScript's HD system boosts antibody expression 2 to 5-fold, as shown in panel B. We also found that in the presence of cocktail 1, both cell density and cell viability were improved by 16 and 17% on average compared to our competitor, and this is shown in panel C and panel D. When we compared our HD system to our competitors, we also found that antibodies expressed in the HD system showed comparable binding affinity and similar glycosylation profiles to that of our competitor. And in particular, for many of our clients in the early discovery phase, they found that our high-density 293F expression system allowed them to take full advantage of high-yield and cost-effective protein production, and was an excellent replacement for their existing expression system. GenScript also offers popular high-throughput gene-to-antibody service that allows researchers to express 4 or more antibodies in small quantities for further testing. It is a flexible service where you can choose to express different types of full-length IgG, as well as other fragments fused with the constant region, such as single-domain antibodies, SCFVs, etc. Here, I just wanted to show that we expressed 8 recombinant antibodies through the system and purified them in a high-throughput format to give And these SDS page gels are just to give you a sense of the quality of our HTP gene to antibody production service. We are also able to produce antibodies and proteins at gram levels. So this is just an example here where we showcase gram level recombinant antibody expression using our high density 293F expression system. Panel A and Panel B show the SDS page and Western blot results respectively. And the purity characterized in Panel C by HPLC is shown to be close to 99%. So as I wrap up this presentation, I hope that I have shown you how engineering your perfect protein or antibody is totally within your reach. At GenScript, we have a talented team of R&D scientists and PhD level project engineers, project managers who are here to help. And just as a summary, I'd like to review some of the key services we offer, but for a full list, please do visit our website at www.genscript.com. For our lead generation services, we offer a comprehensive array of highly customizable antibody generation services. Remember that we are recruiting beta testers for our single B cell sorting service. We do offer express antibody generation services and specialized service like custom ELISA kit development and antibody labeling. For our lead optimization services, it goes without saying that we are a one-stop solution for DNA synthesis. And we also have cutting-edge services like precision mutant libraries that I've shown today, as well as combinatorial DNA assembly. And for protein, we have the high-density mammalian transient expression platform suitable for protein expression at different scales. And while I did not cover it today, we do have protein expression services across the bacteria, yeast, and insect expression systems. And with that, thank you very much for your attention. I'm happy to take questions.
Thank you for Dr. Grace for the presentation. So right now we'll be answering those questions that you have. So the first question, just hang on for a minute. So somebody is asking, he wanted to know more about the mixture that enhances the development of the antibody.
Yes, so these are proprietary cocktail mixes that we use. They contain proprietary growth, growth factors and other stuff that stimulate cell growth and metabolism. And it's been a mixture that's been optimized by our research and development team. And in such a way that, they've compared it to our other competitors and they found that in general, our protein production is, shows better performance. So yeah, so it's just a proprietary mixture that our production team has come up with that really accelerates cell growth and performance.
All right, thank you. The next question, how many antibodies can you produce by high throughput gene to antibody, and what can I use them for?
Yeah, sure. So our capacity is that we can generate about 500 antibodies per week. And these antibodies are usually pretty good for running pilot test assays. So scientists really enjoy this service for screening and binding assays when they have a lot of leads in the beginning. And it's also pretty good for cell-based assays as well.
All right, thank you. The next question, what type of sequences do you accept?
I think you are referring to antibody sequences. So For our
Yes.
protein services, our high throughput gene to antibodies services, we generally accept any antibody system sequence you have obtained. Usually these are, the variable regions sequenced from hybridoma cells, the hits that, people get from display libraries, sequences from single B cells analysis. And so for any antibody variable domains or fragments, we usually fuse them to an FC tag for you. Yeah.
All right, thank you. The next question, what kind of deliverables that you can provide for your mutant libraries?
So for mutant libraries, we offer either double-stranded DNA, or we can clone the variants into your vector of choice. For the site saturation mutagenesis libraries, we usually deliver the variants from each site as individual subpools, or we can also mix the variants together from all the mutated sites in a single mixed pool. We also we provide double-stranded DNA libraries starting at 1 microgram or at 4 micrograms for cloned libraries. If more plasmid is needed, we also have plasmid prep options available for scale-up.
Okay, thank you. So the next question, how many antibodies can I expect to get out of an antibody project?
So this is a trick question. So it really depends on your needs and the antibody generation technology you're looking to use, and it also depends on the animal species that you're working with. So we do have some monoclonal antibody packages that can deliver a set number of hybridoma cell lines or antibody sequences, and that can range between anywhere from 2 to 10 clones. But actually, most of our antibody packages are highly customizable, and we really do our best to deliver as many clones as you need, especially if you want more for screening. And one person might just need 1 antibody for an IHC, but someone else might need 100 antibodies, for leads that they're looking to optimize for drug discovery. So at GenScript, we do have dedicated project managers, and really what you can do is really discuss your project with them, and they will work together with you to assess the scale of your project, how many supernatants you need, and ultimately how many antibody clones you hope to have. So it really depends, but we can definitely tailor our services to your needs.
All right, thank you so much. The next question, could you please explain more about CDR grafting and oligo pool?
Ooh, so that's an awesome question. I will perhaps call on my colleague Kimberly Arnold to help respond to that question regarding CDR grafting and oligo pools.
Yes, hi. I'm Kimberly Arnold. I'm a product manager for our library services. So I'm not too familiar with CDR grafting. If you mean CDR mutants in the CDR region, we do have a number of our mutant libraries do cover our CDR regions. Particularly, a lot of combinatorial and mutagenesis libraries will cover our CDR region, so you can mutate multiple CDRs, so you can do heavy chains at the same time as well. So that could help actually look at simultaneous mutations in CDR regions at one time. So we utilize our oligo pool, our semiconductor technology that Grace talked about to synthesize the mutant oligo variants, and then stitch them together with the constant region for the full antibody sequence. So we can just do the CDR regions if you would like, or we can do the full antibody sequence.
All right, thank you, Kim. So the next question, how is antibody affinity maturation achieved in terms of CDR region? And how do you identify amino acids to be replaced or mutated?
I'll take that question as well. So for affinity maturation, so in terms of designing which mutant sequences you would like, we can help with some of the design, but typically you will provide us with the mutation design that you would like, and the different positions that you would like to mutate, and to which specific amino acids you're looking for, as well as which codon table you would like for your host expression interest, because we can also optimize the codons as well. And so we provide the library. And in terms of Affinity maturation services, we do have additional options as well that we could kind of bundle with a library. But that would be something we can discuss further with our project management team.
All right, thank you, Kim. So the next question, do you employ in silico screening of the antibodies? But if you do, what kind of screening it is?
In terms of in silico screening, do you mean like by ELISA or by fax? If so, we do those forms of screenings as well.
Okay.
Yeah.
Okay. So the next question, can you please describe more about the differences in reducing and non-reducing conditions in HTP formats?
So basically, when you reduce, when you run the reducing gel, you basically are able to denature the antibody, and therefore you see the two different chains, the heavy chains and the light chains for the antibody. And so that's in the reducing conditions, whereas in non-reducing, you see just the antibody as a whole.
Okay, thank you. The next question, do we offer epitope mapping service?
Yes, we do.
Okay, thank you. So for the next question, what do we need to provide for protein expression except the gene?
Yeah, so basically, you know, that's really all you need to provide. We can do cloning into the expression vector for you. So you really just need to you really just need to provide us with the sequence, and we are able to do the codon optimization for you. And basically, there are a bunch of bioinformatics optimization that takes place before, and then if our our production and R&D team sees that there's going to be not much issue with your sequence, and they think that it's a good candidate and for high success rate in expression, we can move forward with that. So you really just need your gene sequence.
All right, thank you. So the next question, do you offer direct sequencing of antibody genes from beacon sorted single B cells?
Yes, we do. So the once the cells are sorted, we do put them through sequencing, and at the end of the day, the antibodies can be recombinantly expressed from the antibody sequence that we obtained.
Okay, thank you. So the next question, he wanted to know more about the mixture that enhances the development of the antibody.
I think this was one of the first questions, and it's a proprietary mixture of growth factors and different things that we can't really share with you. But basically, it enhances the cell's viability and and also enhances expression of the specific antibody formats that are available that are, I guess, linked to each cocktail. So there are two different cocktails, and each one is favored for certain antibody formats.
All right, thank you. So the next question, what are the major problems faced during the scale-up of antibody?
So I think that's a good question, and usually that's what the high throughput gene to antibody or small scale expression services are for. So it's kind of a way that you can gauge to see how how successful your protein will be in being expressed in the first place. So you can start at small scale, and from there, usually R&D can tell based on the success of the expression levels whether it will continue to perform well for scale up services, or whether the conditions that they have established will translate into a scalable option. So yeah, I mean that's all I can really tell you, but I can certainly provide you with more information on the challenges by email separately if needed. Yeah.
Okay, thank you.
Mm-hmm.
So the next question, do you do antibody humanization or chimera generation?
Yes, we do.
Okay.
We do have, just to mention that at GenScript, we do have a biologics research and development team. So, you know, we can generate reagent antibodies, but we also can help you do more humanized antibody projects as well.
Okay, thank you.
Mhm.
The next question, in the silico chip, do you design cDNA corresponding to the Fab region or CDR only, or are you directly synthesizing the oligopeptides using this technology?
Kim, would you like to take that question?
Yeah, so with using our semiconductor chip, we directly synthesize the oligonucleotides up to 170 nucleotides. So using a computer software, we control the addition of each individual nucleotide on the growing oligos. And with our chips, we can create up to 92,000 oligos simultaneously.
Okay, thank you. The next question, do you have an effective way to increase the binding affinity of an existing antibody?
I think that would be mainly through screening. So, you know, you would initially screen panels of antibodies against the target epitope, and basically you would pick out the most ideal binders, and then you would sequence them and sequence the CDR region and kind of gauge from the sequence of the CDR region which areas you might want to mutate, and then basically it's an iterative process. So then you would, you know, re-express the antibody and screen them again through, you know, various binding assays. It could be by ELISA, it could be by SPR. So there are various ways, and we do have those technologies available for us at GenScript for screening.
Okay, thank you.
Mhm.
So the next question, what do we basically mean when we say hotspot degradation removal in antibody engineering?
Kim, do you want to take that question?
Yeah, I'm not too familiar with hotspot degradation, but it has to do with certain motifs within the CDR region, which are degradation sites that kind of break down the antibody, from my understanding. Or like affect the overall stability.
Okay, Ken, we can send more information to the person who asked. So the next question, are we doing any of AVTech technology for labeling antibodies, single protein, single biotin, for example?
So yes, we do have antibody labeling services. Definitely AVI tag and all other common tags. And I think if you have any needs, you can reach out to our department and they can basically tell you what our capabilities are.
Okay, thank you.
Mhm.
The next question, do you use in silico modeling and prediction of antibody properties before designing your antibody?
So usually that would usually we can offer some assistance, but that would normally come from the customer's end. So when they come to us, they usually have some idea of the type of antibody they want to design and express. So yeah.
Okay, thank you. So the next question, can you please shortly explain how to engineer gene?
Kim?
I'm sorry, could you repeat the question again? I didn't.
Yeah, sure. Could you please explain how to engineer gene?
Engineer gene?
Yeah.
Not too sure what this question is asking.
It's okay, we can come back to that later.
Yeah, sorry.
It's okay. Yeah, so due to time constraint, right, we are not able to answer all of the questions, but we'll be sure to email the answers back to you guys. So thank you all of you who joined us for this webinar today, and I wish you a good day today. Thank you. Bye-bye.